Hi, I'm Dr. Scott Friedman, and I'm delighted to talk to you about two articles that I featured in this month's Focus feature in the Journal of Hepatology. The first examines an important process in hepatitis C infected hepatocytes that may promote cancer, and the second involves a potential new therapy for something known as sinusoidal obstruction syndrome using an existing drug. So let's dig into the first article first. The study by Akari and colleagues looks at the impact of overexpressing or expressing the hepatitis C virus NS5A gene and its impact on cellular phenotype. Simply put, they expressed the gene product that's normally found as part of the hepatitis C virus and noted that cells that are expressing this gene product begin to acquire features of something known as EMT, or epithelial mesenchymal transition. This is a process wherein an epithelial cell begins to acquire features that are more typical of a mesenchymal cell, like a fibroblast. Now, EMT is known to occur and is a very important natural process during development when tissues acquire increasing specialization. But it's also known that EMT can be a process associated with cancer, wherein adult epithelial cells begin to acquire mesenchymal features which renders them more invasive and therefore more supportive of the emergence of a cancer, for example, in a liver with hepatitis C. EMT is a critical process in many malignancies. Perhaps the best work underlying this conclusion is from breast cancer, but increasingly EMT may also be found in hepatocellular carcinoma. So the study performed by these investigators in France showed that, in fact, when cells express the NS5A gene product, they begin to acquire features of epithelial mesenchymal transition, both in cultured cells, but also in animals, either when cells are injected or supplanted into animals, or when transgenic mice genetically overexpress the NS5A gene product. So the implication is that we know that chronic hepatitis C predisposes to cancer, hepatocellular cancer in particular. We also know that the risk of cancer increases as more fibrosis accumulates. So this finding may continue to draw links between the pathogenesis of hepatocellular cancer and the specific impact of hepatitis C virus on the behavior of hepatocytes. Now, I need to distinguish EMT in this context from EMT that may, in some articles, contribute to hepatic fibrosis. I would have to say that the bulk of evidence argues strongly against a contribution of EMT to fibrosis per se. Genetic models had initially suggested this might be the case, but more recent studies really argue strongly against EMT generating fibrosis as a primary source of scar in the injured liver. Rather, resident mesenchymal cells, either portal myofibroblasts or stellate cells, when activated, are surely going to emerge as the major source of scar tissue in the chronically injured liver. Now let me turn to the second article, which addresses a critical, although relatively rare syndrome known as sinusoidal obstructive syndrome, or obstruction syndrome, and this was previously known as veno-occlusive disease. It's a catastrophic clinical syndrome that occurs typically in patients who have received chemotherapy, wherein the outflow of the hepatic sinusoids become occluded and leads to rapid onset of portal hypertension, rapid onset of ascites, and a difficult clinical course. Animal models have been developed to mimic sinusoidal obstruction syndrome, most notably one developed by Lori DeLev and her colleagues at USC several years ago, in which they inject into rodents a chemical known as monocrotaline. And so the monocrotaline model has emerged as perhaps our best model to mimic sinusoidal obstruction syndrome seen in patients who develop it on chemotherapy. Long story short, using this model, investigators have found that a well-known drug, sorafenib, which has already been approved for use in hepatocellular carcinoma, may actually have some therapeutic benefit in patients, or I should say in animals with sinusoidal obstruction syndrome, and that may presage the potential use of this drug in patients with the syndrome, since there are no treatments currently available. So this is a nice translational model using a well-validated animal surrogate to investigate the use of an existing drug for a catastrophic condition. The finding and the approach also underscore the emerging effort to use existing drugs for new indications. The NIH, the United States major medical funding organization, has specifically sought applications that propose to repurpose drugs of this type for new indications. And this is likely to be a trend that's going to spread around the world, in which we look to the drugs that already have good safety data to ask, could these drugs possibly be used for other diseases beyond those that originally were envisioned? So this is one example where sorafenib, a drug already approved for HCC, could ultimately emerge as a potential therapy for sinusoidal obstruction syndrome through its potential antagonism of a critical receptor, vascular endothelial growth factor receptor, or VEGF receptor 2. So in summary, I've reviewed for you two interesting articles that employ important animal models to uncover new pathogenesis of disease in the case of hepatocellular carcinoma and a potential new therapy in the case of sinusoidal obstruction syndrome.